So thank you for having me at this meeting. I'm Associate Research Scientist at Dr. Akiko Iwasaki Lab in Yale, and I'm gonna share our findings about 6 differences in immune responses that against COVID-19, which we reported a while ago last year. So as you may know, around 60% of deaths from COVID-19 are reported in males. Male bias in COVID-19 mortality in nearly all countries with available 6 disaggregated aggregated data. The risk of death is around 1.7 times higher in males. And at all ages about 30 years old, males have a significantly higher mortality risk. Sex difference in immune responses against infection is reported widely across species, and some examples shown here. Actually, there are numerous reports starting from sea urchins into humans, and for example, even Drosophila have sex difference in immune responses, which is at least partially attributed to the sex differences in the immune responses, innate immune pattern recognition sensor, toll signaling pathways. And of course, it depends on the combination of pathogen and host. But in general, females mount more robust immune responses to pathogens, which is generally also the case in humans. Male patients have higher viral rolls for Hep B virus and HIV. Conversely, females mount a more robust immune response to vaccines, such as influenza vaccines. However, the heightened immune responses in female can sometimes also lead to detrimental immunopathology in infections. Importantly, sex difference in immune responses show dynamic changes in its patterns over the life course. The sex difference in immunity is even present in utero. For example, a double-blind controlled trial of maternal micronutrient supplementation in Gambia, Africa, demonstrated that periconceptual nutrition has 6 differential epigenetic effects and methylation changes on immune-related genes such as interferons or beta defensins. These differences change over time throughout the life course, and these changes are closely related to the changes in sex hormone levels, which are known to regulate immune responses. And it is important to note that the immune system are differentially affected by aging, depending on sex. Aging induces a decline in the proportion of naive T cells that is more prominent in males, and B cells decline with age only in males, while they are maintained in females. More recent studies showed that males have abrupt and drastic changes in the epigenetic landscape of their immune cells between around age 62 to 64, and subsequently, male exhibit an accelerated immunosenescence phenotype that is characterized by enhanced innate immune pro-inflammatory gene expression, whereas such change occur roughly 5 years later in females, which is roughly corresponds to the difference in the lifespan differences between sexes. So how about COVID? This figure summarizes the factors potentially related to sex differences in immune responses against COVID. For example, ACE2 expression is downregulated by female sex hormone estrogens, while TMPRSS2 is known to be regulated by androgens. Importantly, in mouse model of SARS-CoV-1, it has been shown that female mice are more resistant to infection, but estrogen receptor blockade cancels this sex difference suggesting the protective roles of estrogen in females. In terms of viral sensing, the expression of TLR7 is known to be higher in females. TLR7 is on X chromosome, and although one of the two copies of X chromosome in females is silenced through X chromosome inactivation, some critical immune-related genes, including TLR7, can escape from this inactivation. Also, previous studies have shown higher expression interferon regulatory factor 5, IRF5, in females, and higher expression of these genes, IRF5 or TLR7, leads to more robust type 1 interferon response in females, and higher expression of interferon-associated genes in females in general. Now, more and more studies coming out on sex differences in COVID-19 immune responses. But when we started our research on COVID-19 immune responses last spring, there has been only a couple of studies looking into this important issue. So we decided to work on this problem. So this is our study design. We recruited in total 98 patients admitted to Yale New Haven Hospital for COVID. Samples were longitudinally collected every 3 to 7 days. And as a control, we recruited the healthcare workers at the same hospital who were tested negative every 2 weeks. And we examined these parameters in patients and controls, namely viral loads, plasma, anti-S1 IgG, IgM, 71 cytokines and chemokines levels in plasma, immune cell composition in Pvmcs, peripheral blood mononuclear cells, and T cell phenotypes, including surface marker and intracellular cytokine expressions. So in this study, we conducted two different types of analysis. One analysis was total cohort analysis on all the 98 patients, including all time point samples, and statistically adjusting for age, BMI, disease duration, and treatment interventions. So this is total cohort analysis is in number 1. So number 2, the other analysis was a baseline analysis. We analyzed only the first time point samples. So the first time point was roughly about 7 days days from symptom onset. And we analyzed only these first time point samples who had lower disease score, not in ICU, and were naive for any immunomodulatory agents, such as corticosteroids or tocilizumab, as a first sampling. Unlike the total cohort analysis, this is a raw data analysis without adjustment with statistical modeling, but the age, BMI, and disease duration are well matched between sexes. And in this talk, I mainly focus on the result of this number 2 baseline analysis. So first, these are the results on viral loads and antibody levels. As you can see, there was no statistically significant sex differences about these parameters, although there was a tendency for higher anti-S1 IgG in females. Next, we examined 71 cytokines and chemokines levels in the plasma. The left panel is a heat map of the results, and as expected, patients had generally higher levels of inflammatory cytokines and chemokines, but most of them were without sex differences. However, we found the significant differences in these cytokines. Namely, males have higher levels of IL-8 and IL-18, which have been reported to be associated with severe disease in other studies. Next, we analyzed immune cell composition in Pvmcs. So T cells were decreased in patients as previously reported, but there was no sex difference in the baseline analysis. On the contrary, we saw a marked increase in monocytes for both sexes, reflecting the activation of the innate immune system. And in particular, a subset of monocytes, non-classical monocytes, was significantly more abundant in male patients. These non-classical monocytes have higher potential for transendothelial migration and higher potential for promoting neutrophil adhesion and recruitment. So higher IL-8 and higher IL-18 levels and more robust induction of non-classical monocytes in males collectively suggested more robust activation of innate immune system in males. Then we examine the T cell phenotype in detail. We examine T cell subpopulations such as regulatory T cells, memory T cells, and follicular T cells, and also their cytokine productions such as interferon gamma, TNF, IL-6, IL-17. About many of the T cell subpopulations, the compositions were largely similar between males and females, but what we found differences in some important cell populations. So what we found was a stronger induction of activated and differentiated T cells in females. In female patients, T cells positive for CD38 and HLA-DR, classical T cell activation markers, as well as PD-1 TIM3 positive T cells, differentiated T cells, are more robustly induced compared to male patients, suggesting stronger T cell responses in female patients at the baseline. So far, I exclusively talked about the baseline analysis with only first time point samples from moderate patients without any immunomodulatory agents. But I just briefly mentioned here in this one slide on the result of longitudinal analysis with total cohort around 100 patients. In this analysis, patients' age, BMI, days from disease onset, treatment status are all adjusted statistically. So this is kind of an overall difference throughout the disease course. And with this analysis, we found higher interferon alpha 2 levels in females. So the positive value means higher in females. We found higher interferon alpha 2 levels in females and higher CCL5 levels in males and higher T cell number in females. Then we go back to the baseline analysis. And finally, we wanted to know which immune factor in the baseline are correlated with the subsequent clinical deterioration. We divided patients to two groups, stable group and deteriorated group, depending on the clinical trajectories of each patient after the first time point sample collection. So the sizes for these subgroups are very, very small, but the outstanding finding was that for males, the deteriorated group had higher age and significantly less T cell activation, while in females, rather high innate immune cytokines are related to the clinical deterioration following the first time point samples. So we made the correlation heat map between clinical deterioration and immune markers that we examine, such as T cell responses and cytokine levels for each sex. And as I mentioned in the previous slide, we found that in males, clinical deterioration is correlated with higher age and lower T cells and lower T cell response. Whereas in female, it is not the case, and deterioration was correlated with higher innate cytokines. Then we plotted the age of the patients and T cell activation and found that in males, T cell activation significantly decreases with age, but not in females. In females, T cell activation is maintained up to older ages, which suggests that females age better in terms of T cell responses against SARS-CoV-2 infections. So this is a summary of our study. Male patients have higher plasma levels of IL-8 and IL-18 compared to female patients at the baseline. So in the longitudinal analysis, female had more higher levels of type 1 interferon alpha 2 levels. And at the baseline, male patients have more abundant non-classical monocytes, whereas female patients had more robust T cell responses. Clinical deterioration in COVID-19 is associated with poor T cell activation in males, while higher innate immune cytokine levels in females at the baseline was correlated with the deterioration in females. And importantly, T cell activation significantly declined with age in male patients, but not in female patients. So future directions, since the study size of our study was small, studies with larger cohorts are awaited. But in line with our findings, multiple studies have shown higher higher innate cytokine levels and higher neutrophil to lymphocyte ratio in male patients. Also, studies with single-cell RNA sequencing showed that higher IL-18 expression in monocytes and nasal epithelial cells in male patients. Another single-cell RNA sequencing study showed that 6 difference in mucosal-associated invariant T cells, innate-like T cell subpopulation phenotypes. So male mite cells have more pro-apoptotic gene phenotype, whereas female mite cells have more activated gene phenotypes. These are interesting findings. And correlations between sex hormones and immune responses are a very interesting topic. So it has been reported that menopause as the independent risk factor of severe disease in COVID, and estrogen E2 levels at luteal phase inversely correlate with IL-6 and TNF. Also, another important clinical observation is that androgen deprivation therapy for prostate cancer reduces the risk of severe disease in COVID. It is also interesting to look at vaccine response sex difference, and also important to look at sex differences in long COVID. So a very recent paper published in Nature Medicine a couple of weeks ago showed that increasing age, BMI, and female sex are associated with long COVID. It is also interesting to look at correlation with other factors with sex differences and immune response sex differences. So this is something that we collaborated with Metabolism Lab, and we found that sex difference in tryptophan metabolism correlates with immune response in sex differences. And also, biological sex difference is closely intertwined with social, behavior, and demographic factors, namely gender factors. So these points are important to look at. So finally, there are so many people involved in this huge collaboration work, and I acknowledge all the people involved in this study, your impact team, and fundings. And I'm happy to take any questions.
Thank you. Really good presentation. I think I will start with a question. I'm curious if you have followed up the patients to discover the relationship between sex difference in immunology response and the death rate of the COVID-19.
Sorry, I couldn't hear the last part of your question. So
It's did you follow up the relationship between sex difference in the uh, immunology responses? For example, inflammation and the death rate.
that's right.
Yeah.
So I mean, about the death rate, that's something that we didn't address directly in this study.
Uh-huh.
And yeah, it is very interesting point to look at the long-term difference about the immune response sex difference.
Mhm.
And especially interesting point is, for example, antibody levels differ or not between sexes. So there are several studies concerning these points. And so in general, females mount more robust immune responses against vaccines, for example, against flu vaccines or something. So it hasn't been, so probably lots of studies will be coming out soon, but so far it has been not so many studies concerning, for example, if there is a sex difference in antibody response against COVID. Some studies showed that in convalescent patients, for the severely ill patients, um, a paper showed that conversely, like male had higher levels of anti-S1 IgG levels, but um, it is not known. So, for example, the study only contained severely ill patients. So it has been like not well characterized what happens for like general population with milder diseases. So it is definitely interesting and important things to look at.
Okay, thank you.
Everyone else, feel free to ask questions too. I think with the manageable audience, you can just unmute yourself. I can quickly follow up. So actually, I didn't know of this, but are the vaccine data and efficacy data reported in a sex separated manner? And how does that look?
Oh, vaccine efficacy.
Yes.
Um, uh, as far as I know, many studies have reported the efficacy or results in six aggregated manner. So that's the issue. So that's why we have to emphasize the importance to look with the six different disaggregated manner. So as far as I know, mm, probably most of the studies are reporting the results as six aggregated manner. So yeah, that's important things to look at, I think.
Okay. Thanks. And I see Joanna raised their hands.
Hi, thank you for a very nice talk, and the data was very interesting. And you mainly focused on what is the difference between the COVID patients in males and females. But I was wondering, did you control for like how severely ill people were? For example, if you have a male patient who is also mechanically intubated, and a female patient also mechanically intubated, and given because you showed the differences in the plasma IL-8 levels, that I wonder if even at the same same severity of the disease, do you see any differences between males and females?
You mean, for example, if males and females, some subset of patients have similar levels of inflammatory cytokines, is there any clinical
Yeah.
difference?
Yeah.
That's a very interesting point. We haven't looked at. So the majority of the results I talked about in this talk is about baseline analysis with moderate disease severity. So they only needed like nasal cannula, oxygen supplementation, not intubation in ICU. And yeah, the disease severity were largely comparable. So in the longitudinal analysis, we adjusted or corrected for like treatment intervention and disease severity. So there was statistical adjustment. And as I have briefly mentioned, there was a difference in type 1 interferon levels and CCL5 levels. But yeah, we didn't address that question like directly. So it might be important. Is same levels of cytokines and chemokines meaning same thing for both sexes? It's kind of interesting question.
But do you now think that that question can be addressed with the data that you already have?
Yeah, possibly, yes. Our cohort only contained about 100 patients, and many of them are moderate patients. So yeah, we might need more N, but we can definitely look at. Yeah.
Thank
Thank
you.
you for the suggestion.
Tommy asked, are there interaction between sex and comorbidities like obesity in terms of the immune response?
Yes, that's very, very interesting point. And in supplementary data of our paper, we look at if there's a demographic difference associated with sex difference. For example, if there's any difference in the comorbidity between sexes. And in this cohort, there's no difference, statistically significant difference. But as I have said, like sex difference is also closely intertwined with demographic difference between sexes. So this is very important point to look at.